Hi there, I'm Brenda. And I'm Mia. And today we will guide you to the fascinating world of sequence alignment and phylogenetic analysis using cutting-edge computational tools. Throughout this video, we will explore how biological information like protein or DNA sequences can be compared, aligned, and interpreted. Using advanced mathematical concepts such as Fourier transforms, correlations, and convolution.
Our main focus is to understand how the software MAFFT uses these techniques in order to perform multiple sequence alignments efficiently, and how underlying physical and chemical properties such as volume or polarity of amino acids influence biological similarity. Think of it as speed dating, but for proteins. Hey, you look like you have similar polarity. Wanna align?
No.
Phylogenetic analysis is the study of evolutionary relationships between species, genes, or proteins. And it has some fundamental steps, and we're gonna explain it with a phylogenetic tree of insects. We start by collecting genetic sequences from different species of insects. These sequences are public in databases like in National Library of Medicine. You look for the name of the species, then look for the nucleotide, and then find the sequence to analyze. Next, we perform a multiple sequence alignment using MAFFT. This aligns similar regions across all sequences. We then apply an evolutionary model to estimate how the sequences have changed over time through mutations. Based on the aligned sequences and the evolutionary model, then we construct a phylogenetic tree. This for visualizing how the insect species diverge from common ancestors through time. Yes, it's like a family tree, but instead of grandma, we've got 400 million year old winged ancestors. By analyzing the tree, we can trace some of the most important moments in insect evolution. For example, insects first emerged around 479 million years ago. Later, about 406 million years ago, some of them evolved wings, a huge leap in their evolutionary success. During the time of the dinosaurs, insects with complete metamorphosis like butterflies, beetles, etc. began to evolve and spread. This event is often called the Big Bang of insect diversity. Imagine, while dinosaurs were doing dino things, butterflies were plotting their world domination.
Multiple sequence alignment allows us to compare the sequences and identify homologous regions, that are regions that share a common origin, which allows to understand their evolutionary relation and predict how specific genes behave in relation with different genes. You see, without alignment, it's like comparing different languages without a dictionary. It's confusing, misleading, and probably ends up with someone crying. This study is important not only for understanding gene structure, but also to assign them roles in terms of treatment and more viable therapies for difficult diseases. The purpose is to improve the finding of sequence similarities and to have a mechanism for bioinformatic innovation.
MAFFT stands for Multiple Alignment using Fast Fourier Transform. Its innovation lies in how it translates biological sequences into numerical data and uses Fourier mathematics to accelerate the process. Each amino acid is assigned numerical values based on physical properties, and instead of comparing characters directly, MAFFT transforms the sequences into frequency space using Fast Fourier Transform. In frequency space, patterns and similarities are easier and faster to detect. MAFFT detects highly similar regions and proposes alignment, and then refines them. This approach allows MAFFT to align thousands of sequences in a fraction of the time other methods will take, without sacrificing quality.
The use of linear convolution allows for improving the correlation computation, making it more efficient to identify sequence similarities. The number of amino acid sequence expressions is related to the difference between physicochemical properties of both amino acids, mainly polarity and volume. To make meaningful comparisons, it's necessary to normalize the data corresponding to these properties, which is accomplished by subtracting the average of the group of amino acids from the individual value and then dividing it by the standard deviation. Using the normal volume and normal polarity, the correlation of each property leads to the correlation function. The CK mathematical function is a correlation between two amino acids and their similarity when shifting one sequence by k positions. The high value of CK means that the sequences may have homologous regions. If two sequences have homologies, the correlation CK has peaks, as shown in this figure, where we observe a result of the Fast Fourier Transform analysis. There are two peaks that correspond to two homologous blocks. This mathematical function is similar to the correlation coefficient proper of statistics, as in both correlations, a set of values for data points is used in order to determine the strength of a relationship between two variables.
Understanding tools like MAFFT and concepts like normalized volume, polarity, convolution, and correlation allow us to grasp more on computational biology and how it translates the language of life. It's like Google Translator, but for evolution.
We hope you enjoyed this deep dive into sequence alignment and phylogenetic analysis. Thank you for watching, and remember, science is all around us. We just need the right tools to see it and probably some math magic.